Hello friends, welcome back to NitVed Bioscience. Today we will discuss about the oxygen binding curve for hemoglobin and myoglobin. So before starting the actual topic, let us revise the few things about the hemoglobin and myoglobin. Hemoglobin, it is a tetrameric protein having the 4 globin chain and the 4 heme group is present here, and myoglobin has only the single polypeptide chain or the globin chain, and the single heme group is present in the myoglobins. Hemoglobin, it is a transport protein whose main function is to the transport of oxygen from the lungs through blood and to the tissues, various cells and the tissues which is in the need of the oxygen. So it is a transport protein which is present in the RBC. Myoglobin, it is a storage protein whose main function is the storage of oxygens, and when this hemoglobin will not be able to supply the oxygen efficiently or effectively, in that case this myoglobin will supply the stored oxygen. So these myoglobins, from the name myo means muscles, these are the storage protein in muscle cells. As it has a 4 polypeptide chain, so hemoglobin shows the principle of the cooperative binding that we had discussed. What is cooperative binding? Cooperative binding means there are the 4 polypeptide chain or the 4 globin chain, right? So binding of oxygens to any one of the globin chain will make the conformational changes in such a way that the affinity of other poly or other subunit will increase towards the oxygen. Okay, binding of the one subunit to the oxygen will make the conformational changes in such a way that so affinity of other subunit will increase towards the oxygens, and then other subunit will more readily or rapidly will bind to the oxygen. So that is a cooperative bindings. This principle or this phenomena we will see in the hemoglobin, cooperative binding. But as the myoglobin has only the single polypeptide chain, so there is no cooperative binding. Okay, so hemoglobin shows the phenomena of cooperative bindings, while the myoglobins no cooperative binding as it has only the single globin chain. Hemoglobin has a low affinity towards oxygen. Low affinity, while the myoglobin has a high affinity. Meaning of this is that myoglobin will bind to the oxygen more tightly as compared to the hemoglobin. Therefore, it is binding to the oxygen more tightly. Therefore, it is a storage protein and it is a transport protein. Because it is not binding the oxygen as tightly as myoglobin. So, it can easily pick up the oxygen from the lungs, travel through the bloodstreams, and will supply to the organs or the tissues which is in the need. So this shows the low affinity towards oxygens or the loose binding as compared to myoglobins. These are the some things which we need to keep in the minds before understanding this topic, oxygen binding curve for the hemoglobin and myoglobin. Okay, now there is one more things which we need to understand is the partial pressure of oxygen. Partial pressure of oxygen. So, suppose let us first understand this partial pressure of oxygen by taking just example of hemoglobin. Okay, so hemoglobin has a 4 globin chain, means it is a 4 oxygen binding site is present here. Partial pressure of oxygen we can denote by PO2. So partial pressure of oxygens. There are many hemoglobin molecules. Just for our understandings, we are taking just one hemoglobin molecules, and it is a 4-seater transport vehicle. Suppose this is a beaker where the mixture of hemoglobin is present. In our blood, in the human, we know the level of hemoglobins, right? So for male, it is like around the 15 gram per deciliter, and for female it is around the 10 to 12. Female has a low hemoglobin as compared to the men's. So here suppose standard we can say that 30 to 15 gram of hemoglobin is present in this mixture, and this is some beaker, and we apply the pressure here. Sorry, my mistake. We will apply the pressure in these directions, and here the dissolved oxygen is present. Okay, oxygen is dissolved in the mixture. So here is a beaker. In the beaker, just for our understanding, okay, so in a beaker, hemoglobin, mixture of the hemoglobin is there, and the oxygen is also present here, and we are applying the pressure by a piston. Okay, so if the pressure is more, oxygen will bind to hemoglobin, or the association or the binding of oxygen will take place on the hemoglobin molecules. So oxygen will bind to hemoglobin. This phenomena or this reaction or this binding takes place in the lungs. Okay, in the lungs, when the partial pressure of oxygen is more, which is 100 mm of Hg. This is the unit of the partial pressure, millimeter of mercury. So in the lungs, where the partial pressure of oxygen, and the partial pressure of oxygen is always calculated by dissolved oxygen. So the dissolved oxygen partial pressure is more, so association or the binding of oxygen will take place on the hemoglobin. Okay, and then it will travel through the blood streams and ultimately reach to the tissues. Okay, ultimately will reach to the tissues. In the tissues, there can be of two types, if it is a resting tissue or exercising tissue. Okay, so in both the case, the partial pressure of oxygen is less as compared to lungs. In the lungs, partial pressure of oxygen is more, so the binding takes place. In the tissues, resting tissues or the exercising tissues, even less. In the exercising tissues, partial pressure of oxygen is even less as compared to the resting tissue. So there the dissociation will take place, and that oxygen will unload. Oxygen will unload from the hemoglobin, and then oxygen will be supplied to the tissues or cells of the tissues, and which is ultimately used to generate ATP. So what my point here is that in the lungs, when the partial pressure of oxygen is more, so binding will take place, then it will travel. In the tissues, partial pressure of oxygen is less, so dissociation. Association in the lungs, dissociation in the tissues, because of the difference in the partial pressure of oxygen. So that things we need to understand here. Partial pressure of oxygen we can denote by PO2. In the lungs, the partial pressure of oxygen is 100 mm of Hg. PO2 we'll see at the three different levels. Means in the first in the lungs, then in the resting tissues which is not exercising, and the third is the exercising tissue. Exercising tissues. So in the lungs, partial pressure of oxygen is 100 mm of Hg. In the resting tissues, this partial pressure of oxygen drops to 40 millimeter of mercury, and in the exercising tissues, okay, exercising tissues, it is a 20 mm of Hg. We'll discuss this value in detail after some times when we discuss the graphs, but you just understand the partial pressure at the various level is different. In the lungs it is more, in the resting tissue it is less, and even less in the exercising tissue. So this will favor the loading and the unloading of oxygen from hemoglobin. But when the case is even more demanding, when the enough oxygen is not there, in that case myoglobin will come into action, and that stored oxygen then will supply. Okay, so first we understand why hemoglobin is a more preferred transporter or the carrier of oxygen over the myoglobin by using these values. Again, we'll see the partial pressure. So, in the lungs for hemoglobin and myoglobin. Okay, resting tissue and exercising tissue. Okay, we will see the values of how much oxygen will be unloaded under various partial pressure by hemoglobin and myoglobin, and then we will see the graph. When we apply the pressure which we had discussed in the lungs, the partial pressure of oxygen is 100 mm of Hg. At that time, the hemoglobin which is present in the mixture, it is almost 98% saturated. Means out of 50 gram, 98% of the 50 gram is saturated or the loaded with the oxygen. Okay, and as we move on to the tissues or the exercising tissues, so unloading will take place, and that percentage we will discuss. So in the lungs, at the partial pressure of 100 mm of Hg, saturation of protein with oxygen, saturation of protein means oxygen means how much percentage of the proteins are saturated, fully saturated with the oxygens. So that is the fractional saturation of protein. Okay, two terms we need to understand, and that is by using these two terms we will plot the graph of oxygen binding curve. That curve we will have the two values. One will be the partial pressure of oxygens and the other value will be the fractional saturation of the proteins, means in a mixture of the proteins how much percentage of the proteins are saturated with the oxygen. So in the lungs, when the partial pressure of oxygen is 100%, means 100 mm of Hg, so 98% of hemoglobin is fully saturated with the oxygen, and even the myoglobin also, 98% of the myoglobin under the partial pressure of full partial pressure of 100, means in the lungs 98% of oxygen 98% of myoglobin is also fully saturated with oxygen. So myoglobin also fully saturated and hemoglobin also saturated. As we move on to the resting tissues, in the resting tissue, as the partial pressure becomes the less, okay, here it is 100. So now from the 100, the partial pressure drops to 40. As the partial pressure will drop, so that protein or hemoglobin will supply or unload oxygen. So how much oxygen will be unloaded, that will calculate. So 98% is saturated. At the resting tissue, we will get the 77% will be saturated now. 77% saturated. Earlier it is 98%, now it is 77%. So 98 minus 77 means 21% will be unloaded. This much oxygen will supply to the tissues and 77% will be saturated here. Okay, earlier all the hemoglobin molecules are fully saturated, then 77% saturated. 77 saturated means 21% hemoglobin protein has supplied the oxygen to the tissues. And further, if the exercising tissues which has a more demand of oxygen, because as it is exercising, so exercising tissue, so here the need of ATP will be more, so oxygen demands will be more. So now the percent saturation will further decrease here, and now in the mixture that we have seen, which is earlier it was fully saturated, in the exercising tissue we will get only the 32% saturated. 32% saturated means 66% unload. 66% protein has unloaded the oxygen and supplied to the tissues. So sharp fall in the percentage of unloading or the supply of the oxygen we will see here. So earlier 98% in the resting tissue where the partial pressure is 40, so 21% will be unloaded, 21% oxygen, 21% of the protein will supply to the oxygen, and then 66% will supply to the oxygen here. But what about the myoglobin? In the myoglobin, when 100 mm of Hg, so 90% myoglobin will be saturated. In the resting tissue, when the 40 mm of Hg partial pressure is there, so only 1% of myoglobin protein will supply oxygen, and that is very less. Here we will see that 21% will supply to the resting tissue, and only the 1% myoglobin will supply at the 40 mm of Hg partial pressure. Again, the partial pressure will drop in the exercising tissue to exercising tissue. Sorry, I have not mentioned here. Exercising tissues has a partial pressure of 20 mm of Hg. Sorry, that's my mistakes. I have not mentioned here. Lungs have the partial pressure 100, resting tissue has a 40, and the exercising tissue has a 20 mm of Hg partial pressure. So at the 20 mm of Hg partial pressure, only the 7% of oxygen will be 7% of the protein will supply the oxygen. So that is also very less, and that will not meet the demand of exercising tissue, because here hemoglobin is supplying the 66% and the myoglobin supply only the 6% oxygen. Therefore, hemoglobin is used as a transporter protein by our body. Okay, and the myoglobin will use as a stored protein, because when the partial pressure even drops to very low, right now exercising tissue is a 20, but when the partial pressure becomes very low, almost about the 2 mm of Hg, at that times there will be the sharp fall in the percentage saturation of myoglobin, and all myoglobin will rapidly supply the oxygens all together. Okay, all myoglobin will supply the oxygens all together. So that will be at the very low partial pressure of 2 mm of Hg. Until that times, means this is the need. When the hemoglobin is not able to supply, at that time myoglobin will supply. Otherwise, this value is very low. Therefore, it is used as a stored, and this is used as a transport of oxygen. And depending upon this, now we will understand the curve. So this is the oxygen binding curve for hemoglobin and myoglobin. This first curve, upper one, this hyperbolic curve, it is for myoglobin. This is for myoglobin, and this is for hemoglobin, HB. Okay, upper one is for the myoglobins. For myoglobin, we will get the hyperbolic curve, and for hemoglobin, we will get the S shape or the sigmoidal curve. Okay, this is also known as the oxygen binding curve or oxygen dissociation curve. Okay, suppose going this way in this direction from here to this way, so that is the oxygen binding curve, and this direction it is the oxygen dissociation curve. So we can use this graph interchangeably to understand the oxygen binding and oxygen dissociation. Okay, hyperbolic curve, it is for the myoglobin, and this sigmoidal curve is for the hemoglobins. X-axis is the partial pressure of oxygens, and the y-axis is the fractional saturation of protein, that is ranges from 0 to 1. 0, 0.1, 0.2, 0.3 means 0 means none of the protein is saturated with the oxygens, and 1 means 100% of protein is saturated with the oxygen. So 10%, 20%, 30%. So it is in the percentage. Okay, now let's start talking about this curve first with the myoglobin. Okay, first with the myoglobin. Myoglobin has only the single globin chain. One. Second, no cooperative binding. Third, higher affinity towards oxygen. So because of these properties, as soon as we begin to increase the partial pressure even at a very low amount, all the myoglobins will bind suddenly because there is no cooperative binding. So all the myoglobin will bind because it has a higher affinity. So even at the very low partial pressure around the 2 mm of Hg, at the value of 2, for example here, at the value of 2, we'll get around the 50% of the myoglobin saturated with the oxygen. Okay, myoglobin has a higher affinity, so even by increasing the partial pressure of oxygen, even by the very tiny pressure or with very small amounts, all the myoglobin will bind together with the oxygens, and we will get the sharp increase here in curve for the myoglobin until it levels off. Okay, and then the will remain the same, means fully saturated. 90%, 98% will be saturated for the rest of the value. So sharp increase here we will see in this myoglobin curve with the very small partial pressure of the oxygen. And so this way we had discussed that it is a binding curve, and at the 2 mm of Hg, 50% of the myoglobin will be saturated with the oxygens. Now coming this way, means as we decrease the partial pressure of the oxygens, so just we had discussed that from 100% in the 100 mm of Hg, 98% of the hemoglobin is saturated, but coming down, so we had discussed the value that in the resting tissue, when the partial pressure of oxygen is 40, only 1% is saturated here. So we can see from the graphs there is not a significant drop in the curve for the myoglobin. So only 1% will be dropped at the partial pressure of 40, and in the exercising tissue that value is 7%. Okay, so only the 7% will be unloaded at the exercising tissue, but when the value drops to a certain value, so there will be the sharp decrease in the curve, or a sharp decrease in the unloading of the myoglobin will take place more rapidly until it reach to the very low value. Okay, again I am repeating, myoglobin has a higher affinity towards the oxygens, so even by increasing the partial pressure with a very small value, there will be the sharp increase and decrease also. In the reverse case, when they be talking about the dissociation, so sharp decrease at the very low partial pressures. For the rest of the times it will remain the saturated, therefore myoglobin is used at the storage so for hemoglobin we had discussed that at the partial pressure of the 40, 77% of hemoglobin will be saturated, means 21% will be unloaded, and this value will fall to very sharp when the partial pressure drops from 40 to 20. At that time 32% will be the saturated, means 66% will be unloaded by hemoglobin. Okay, and this phenomena, this S-shaped curve or the sharp decrease here, or this S sigmoidal shape we will get because of the cooperative binding of the hemoglobin. That cooperative binding means binding at one molecule will increase the affinity, and the reverse will be the case when in the dissociation, if there is a unloading from the one subunit, so other subunit unloading from the other subunit will also takes place rapidly, therefore we will see this sharp fall here for hemoglobin. Myoglobin will get the hyperbolic curve because at the very low partial pressure there is a sharp increase until it levels off, and then the remains at the same value. For hemoglobin, this we will get the sigmoidal curve, means in the lungs it is a fully saturated or 90% saturated, and by coming down when the partial pressure drops, this much oxygen will be the unloaded, means this much oxygen is fully saturated. Okay, so we will get the sigmoidal curve here. So this is about the oxygen binding curve for the hemoglobin and myoglobins. These factors we had discussed depending upon the partial pressure of oxygen, but there are the other factors like 2,3-BPG, or carbon dioxide, and hydrogen ion concentration means pH. So that will also affect the unloading of hemoglobin. For myoglobin we'll not discuss, but for the hemoglobins, because as the hemoglobin is a transporter proteins, so this value will also affect the unloading of hemoglobin. Okay, 2,3 bpg, carbon dioxide and the hydrogen ion concentrations. This value or the discussion about this we will see in the next class. Okay, thank you.